Okay, recording has started. All right, so I spoke about this topic last week in detail, and I said this is a type of cell division. Just before we get into the gist of everything, I said this is a type of cell division whereby one cell is going to divide to form 4 cells, but it doesn't divide once to form the 4 cells. So this cell is going to divide to form 2 cells under meiotic division 1. Then at the end of meiotic division 1, both the cells are going to continue with the division in order to form 2 cells each. And then at the end of meiosis, we will then have 4 cells, and these cells are genetically non-identical. These 4 cells, we say they are, obviously my pen won't write perfectly at all times, but I think this week it's been behaving. Genetically non-identical. Non-identical. That's what we said. And when we speak about meiosis, we also need to understand that per division, there are phases. And whenever you name the phases, you need to include the number of the division. So in this part of meiosis, we say that is meiosis 1. So whatever phase that you mention, prophase 1, metaphase 1, anaphase 1, telophase 1, that 1 needs to be there. Whether you write it as 1 is in a number or 1 Roman figure, that is fine. Then on this part, whereby the 2 cells at the end of meiosis 1 continue to divide, that is known as meiotic division 2. So the names of the phases here need to have the number 2. Anaphase 2, metaphase 2, telophase 2, prophase 2. If you don't have the number, you are going to lose a mark. So I think that is basically what happens with meiosis. And the main goal of this cell division taking place in our cells is to produce sex cells. So sex cells being the sperm cells and egg cells. So egg cells for females and then sperm cells for males. So that is the goal for meiosis. All right. Now, I'm not sure if I mentioned this last week, but meiosis is tested in paper 2, and it has a total of 21 marks. I think you should be aiming to get plus minus 17 from this topic. Plus minus 17 marks in meiosis. Because there are a few elements of challenging questions, but anything that is basic, anything that is asking you to explain or describe, that's something that you should be able to get full marks for. So then we are giving a leeway of about 4 marks to say, if you made a mistake here and there, that's fine. So we've already covered DNA, and we are busy with meiosis, and our next topic will probably be genetics because that's a big topic. Then we'll have to plan around how we're going to do some of the topics also. Now, this terminology is very important. Please try and, make notes if you've printed these notes as I speak about the terminology. I will address some terminology and others I will not address, because that means I will cover it in the lesson. So please find space to write whatever that I'm writing so that you can understand, and please stop me anytime. Now, the terminology that we have here is terminology that we are going to use in meiosis, and some of the terminology we're going to use in genetics, because DNA, meiosis, genetics, there's a link between those 3 topics. Now, the first terminology that we are speaking about here is a gene, and I will continue be saying gene, 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 gene. Please take note of the spelling. It's G-E-N-E, not the jeans that we wear, not the pair of jeans. And it happens a lot at the marking center where learners write J-E-A-N, and you're not going to get a mark for that. Now, what is this gene particular? Right? So this is a section of DNA in a chromosome. You can say it's a section or DNA, or segment rather, of DNA in a chromosome. Now we need to remind ourselves of what a chromosome is. So if I were to take it back here, because I have space here, last week I said this thread here can be regarded as a chromosome, which is made up of one chromatid. Now during DNA replication, this chromatid will obviously be replicated, meaning have a copy of itself. And then we can end up having a chromosome that is made up of two chromatids. So this is the same thing. The only difference is that this is a chromosome made up of one chromatid, and this is a chromosome made up of two chromatids. Now I'm trying to explain the gene. So if I were to draw this chromosome a little bit bigger, so this particular section of a chromosome, so this chromosome contains DNA, and DNA is packed into different sections because a particular section will have a sequence of bases, those bases T, A, C, G, whatever, right? That sequence determines a particular characteristic, and normally it's a body characteristic. So this section of DNA over here can code for eye color or code for hair color or anything that is of a body characteristic. This section of DNA will be known as a gene. So a gene is a segment or a section of DNA in a chromosome that codes for a certain characteristic. So if we're to zoom out a little bit, we can say, okay, in that particular section, these are the bases that we have. So our hair color may be, for example, is determined by this sequence, TACC. Then obviously on the other side, since our DNA is double-stranded, this will be ATGG. This is a section in a DNA which we find in a chromosome that codes for a particular characteristic. This will be another section of DNA that codes for another characteristic, and that characteristic may be hair texture, curly hair, straight hair, or your hair color, blonde, black, brown, red, whatever. This can also be another section. This can also be another section. That is a gene. They always ask about a gene. That is a segment of DNA in a chromosome that codes for a particular characteristic. Then you can even give examples here, e.g., that can be eye color, wow, eye color, wow. Okay, remember this EG is not an example of a gene, it's an example of a characteristic. Okay, then the second terminology is a locus. This is very important. Locus is the singular word for it. Then in plural, it's called loci. Please make a note. I'm not sure what happens in exams. Please make a note that you will be marked wrong if you write locust. There is no T at the end. Locust is something else. We are speaking about a certain animal now. Now, what is this locus? Is a position of a gene in a chromosome. Position of a gene in a chromosome. What did we say a gene was? A section of DNA in a chromosome. Now, this particular section of gene is occupying a certain position on a chromosome. So that means this particular position for eye color, and this is another particular position for hair color. This is another position for blood group. So that means you will find a gene in a particular locus of a chromosome. Check the terminology. You will find a gene, a section of DNA in a particular locus, meaning position of a chromosome. All right. Then let's look at this one, centromere. Oh, the words look and sound the same here. So that's why I say terminology is quite important. Centromere, then we have centrosome, then we have centrioli. Now, centromere is a region whereby the two chromatids of a replicated chromosomes are joined together. So if a chromosome is replicated, it will have two chromatids. This is the first chromatid, and this is the second chromatid. Now we are going to need something that is going to join these chromatids together. It's normally drawn as a big dot or a small dot there in the middle to show that we are joining the two chromatids. So this is a centromere. Centrosome is an organelle. These two go together. A centrosome and the centriole. You can say it's a centriole or centrioli, because sometimes it has an I at the end. Doesn't matter which word which spelling you use, centriole or centrioli, you'll still get a mark. Now, this centrosome is an organelle which will contain the centriole. So centrosome is the organelle which contains the centriole. Now, what we need to understand here is that the centriole, you find it inside the centrosome, and it produces what we call spindle fibers, and we are going to speak about them. During cell division, you will have two centrioles that will move to opposite poles. And when they move to opposite poles, they form spindle fibers between them. So this would be one centriole, also normally represented as a dot in a cell, and another centriole. So when they move to opposite poles, they normally form what we call spindle fibers. So these would be the spindle fibers. This would be a centriole, and this would be a centriole. Then a chromatid. This is one of the two identical strands of a replicated chromosome. So I spoke about a chromosome that is made up of one chromatid, right? Then during DNA replication, you will then get a chromosome that is made up of two chromatids. We can refer to this chromosome as an unreplicated chromosome, meaning this is a chromosome before DNA replication. And then this chromosome can be referred to as a replicated chromosome, meaning DNA replication has occurred. Chiasma. So we will remember chiasma from last week, whereby crossing over takes place and we have homologous pairs of chromosomes. We'll have one chromosome from the mother and one chromosome from the father, whereby they will come in close contact, and chromatids from each chromosome are going to cross over. And where they cross over, that is the point which we call chiasma or chiasmata. Then, autosomes and gonosomes. The terminology here, very important. Sounds similar, autosomes, gonosomes. When we speak about autosomes, we are speaking about the first 22 pairs of chromosomes. Remember, in our human cells, we have 46 chromosomes in total, right? But now, if we are not counting the number of chromosomes, we are counting them as pairs. That means we have 23 pairs of chromosomes. So 23 pairs and 46 chromosomes is 6 and 9. It's the same thing. But when we look at the chromosomes in pairs now, and remember, if it's a pair, that means this one is from the mother, this one is from the father. That is pair number 1. Same with pair number 2, same with pair number 3, and they make up the 23 pairs. Now, from the 23 pairs, the first 22 pairs of chromosomes are called autosomes. And these are chromosomes which will carry genes, meaning DNA, sections of DNA, which will control how we look in terms of appearance, that will control your structure and functioning of your body. So from your blood groups to certain proteins that are formed to normal functioning of the body, all of that DNA is carried in the first 22 pairs of chromosomes. Now, we said you have 23 pairs in total, and the first 22 is regarded as autosome. The last pair of chromosome is known as your gonosome. So 22 pairs, we call that autosomes. Then one pair, which is the 23rd pair, that we refer to as gonosomes. Now, this pair of chromosomes, the last pair, is the one that determines our sex, meaning our gender. So chromosome pair number 23 of the sex chromosome is responsible for sex. Now, in this case, when we mean sex determination, we are speaking about gender, boy or girl, male, female. Now, when we speak about the female gonosomes, we refer to them as XX chromosomes. So any female here will have the XX chromosome, and the male gonosomes will be XY. And we're going to look in detail about this one, I think, when we get to it. Then telomere, this is the end of a chromosome. It protects the genes from damage. So if this is a chromosome, at the end of a chromosome over here, we're just having a telomere there. Then an allele. I'm going to skip this one, and I'm going to skip this one and this one. These three I'm going to skip because they are going to be more important in genetics and not necessarily here. Then germ cell, gametogenesis, all of these, it doesn't help for me to explain them now because these are processes. So it's not just terminology. So I will explain them when we get to that part of the topic. Now we're looking at everything now. DNA, I've covered DNA and chromosomes. Chromosome, very important. They contain DNA. A replicated chromosome consists of two chromatids. This is a chromatid, this is a chromatid, and joining the chromatids together is a centromere. So us human beings, so as much as we are multicellular organisms, we are made up of millions of cells. So if I were to zoom into one of your cells, we should be able to see 46 chromosomes. Please make a note here. You will be required one way or the other to draw the structure of a chromosome, and that could be in the different phases of meiosis. Please note the following. If you draw an X and then you put a dot here in the middle as your chromosome, you are going to be penalized. You need to draw a nice shape of a chromosome. It doesn't have to be perfect, but you're showing that these are threads and then there is a dot in the middle. And X is not going to cut it. Even if your X looks like this. If your X looks like this, and then you have a big dot there, wrong also. You will be penalized for it. So please make a note. You need to learn how to draw the chromosomes. And also they mustn't look like hearts. So if they also look like hearts, something like this, you're not going to get a mark. I think even in the past 3 years, they were not marking it at the marking center. So please make a note. Then we spoke about homologous chromosome pair. And the word pair means two. So when we look at the somatic cells, which are body cells, right? Now, all of these chromosomes in body cells are always in pairs. We find them in pairs. Now, why are we finding them in pairs? We are finding them in pairs because for that particular pair, there will be a chromosome from the mother and chromosome from the father that will determine certain characteristics for that particular pair. Now, why do we call certain chromosomes homologous chromosome pair? They are going to ask you. Those two chromosomes, which we call a homologous chromosome pair, they will need to be the same size. I spoke about this. It will need to be a chromosome from the mother and from the father that will have the same size, and then will have the same length, and then we'll have the centromere in the same position. Check what needs to be the same here. It needs to be the same size, same length with the centromere. Remember, the centromere is what joins the chromatids together in a replicated chromosome. It needs to be the same position. What is also important is that the chromosomes, one from the mother, one from the father, will also need to contain genes from the same characteristic at the same locus. Check terminology. That means they need to contain the same genes for the same characteristic at the same locus. What do they mean by this? So if this is a replicated chromosome from the mother, and this one is a replicated chromosome from the father, this particular position, I'm hoping to change the color here, this particular position in a chromosome. And this particular, remember we said this one maybe from the father and this one from the mother. Even if I swap them around, it's fine. So this particular position of a chromosome and this particular position of this chromosome both need to code for the same gene if it is eye color. So meaning there should be information about eye color here and there should be information about eye color there. So they need to contain genes for the same characteristic at the same position. Same this part over here. If this part codes for blood group, this part should also code for blood group from 2 chromosomes, 1 from each parent. Then there's another terminology, haploid and diploid. Haploid, think half. Diploid, think double. But you're not multiplying anything here. So haploid refers to 1 chromosome from each pair. So you're only having half of the full complement of chromosomes. So if you are saying the total number of chromosomes that you should have in your cells is 46 chromosomes, that is the full complement. But now if we are asking you the half of those chromosomes, that will be 23. So for me and you, the haploid number of chromosomes will be 23. Now a diploid refers to the full set of chromosomes, refers to having pairs of the homologous chromosomes. You're having a pair of chromosome, 1 from the mother, 1 from the father, and you're having the full complement, the diploid complement of our chromosomes, that will be 46. Now, in the notes, this will be referred to as N and 2N. N is for haploid, you have half the number of chromosomes. And then 2N will be for diploid, you have the full set of chromosomes. So how you should also remember this, if I were to make an example of 8 chromosomes as a full complement, so meaning diploid number of chromosomes, that will be 8, and haploid will be half of 8, which is 4. Now, what you are also seeing here is basically 1 copy of each chromosome from the pair. So these chromosomes are not in pairs. So these are just 23 chromosomes, or in our example, 3 chromosomes. Then diploid, you are having full sets of chromosome. These are still 3 copies of chromosome, but they're actually 2 per pair. So this is 1, 2, 1, 2, 1, 2. And these chromosomes are unreplicated. Okay, so now I think I said 3. It should actually be, let's see, 1, 2, 3. So this is 1 copy of each chromosome, 2 copies of each chromosome, 1, 2, 3, 4, 5, 6. Okay. Then diploid and haploid, I can skip this one. I don't think there's anything major that I need to explain. It still has the information about diploid and haploid. Okay, maybe let me explain this. So meiosis takes place in us animals. Meiosis will take place in the gonads in order to form sex cells and egg cells. So meaning in males, meiosis will take place in the testes. So in males, this is a male. So in the testes cells, this is where meiosis is going to take place in order to form a sperm cell. Now, when we look at your cells as a male, we just zoom into one of the cells, whether it's in your skin or in your heart or whatever, it will have 46 chromosomes. Now, from that cell, but specifically testis now, we will then undergo meiosis and then produce sperm cells with 23 chromosomes. So that means we are moving from diploid, which is 46, to haploid, which is 23 chromosomes. Same with females. Meiosis will take place in the ovaries, right? And then when meiosis takes place in the ovary cells will have 46 chromosomes, which is 23 pairs. When meiosis takes place, it takes place to form egg cells. And these egg cells will have half the number of chromosomes, which is 23. Then during fertilization, whereby there's a fusion of a sperm cell and an egg cell, The sperm cell brings in 23 chromosomes from the father, and the egg cell brings in 23 chromosomes from the mother. 23 plus 23 will give us 46. The zygote is the early stages of the embryo, fetus, or a baby. So that means the baby will have 46 chromosomes from the zygote stage, and that is going to be maintained by mitosis. What do I mean by that? When mitosis takes place, remember, it maintains the chromosome number. So any cell division of mitosis that takes place in the cell, it will be a cell with 46 chromosomes, which will divide to form two cells, which will also have 46 chromosomes each. Remember, in meiosis, we are halving the number of chromosomes. In mitosis, we maintain the number of chromosomes. Now, this is your homologous chromosome. Two replicated chromosomes in this case, and we said they need to be the same size, same height, gene in the same locus, and centromere in the same position. So this is a gene for eye color. There it is over here. There it is over here. You're finding it on both sides because remember, this is a replicated chromosome. Then this particular locus, we are coding for hair color. And then this is your centromere. And then this is another gene locus, meaning a position for another gene. Two chromosomes, one from the mother, one from the father. This is still your homologous chromosome pair. How many replicated chromosomes can you see here? You are able to see two chromosomes. Even if I say how many chromosomes can you see, you are able to see chromosomes. This is chromosome 1 and chromosome 2. If a question says how many chromatids can you see, the answer will be 4 because you are able to count the chromatids. This is chromatid 1, this is chromatid 2, this is chromatid 3, this is chromatid 4. How many homologous pair of chromosomes can you see? Only one. This is one homologous pair of a chromosome. This. So I hope you understood my terminology. How many chromosomes can we see? 2. How many chromatids can we see? 4. How many homologous chromosome pairs can we see? 1 pair, because a pair is made up of 2 chromosomes. Now, if you don't want to use the word replicated, you can use the word duplicated. That is allowed. Okay, and remember, from these 2 chromosomes, since they form a pair, meaning one chromosome is from the mother and the other chromosome is from the father, it's still terminology. Let's look at what is happening here. These 2 diagrams, same. This diagram is the same as this one. What's the difference here? The chromosomes on the first diagram are unreplicated. So the chromosomes are made up of only one chromatid. So this is chromosome number 1 and this is chromosome number 2. Then DNA replication takes place. This particular chromosome will make a copy of itself, and this purple one will also make a copy of itself. Then we end up having still 2 chromosomes, chromosome 1 and chromosome 2. What is the difference here? In the first diagram, we are able to see the homologous chromosomes unreplicated. But in the second diagrams, we are able to see the homologous chromosomes replicated. Okay. Then human karyotype. This is terminology. So when we speak about a karyotype, we are speaking about a little diagram that we normally get given in a test. And this diagram gives us the number, the shape, and the arrangement of the chromosomes. So normally this will be in a human cell and we'll able to get a full set, meaning we should be able to see 46 chromosomes. So we are going to see the number of chromosomes, which should be 46, the different shapes of the chromosomes, and the different pairs. They're normally in pairs. So they will be in 23 pairs. We are able to see the arrangement for all of that. Let's remind ourselves of the terminology. From the 46 chromosomes that we have, if we arrange them in pairs, we will get 23 pairs. The first 22 pairs are called autosomes. Then the last pair, meaning one pair has 2 chromosomes. So that's why you are having the word gonosomes next to the number 2. So meaning in total, you are having one pair of gonosomes, meaning 2 gonosomes. Then you are having 22 pairs of autosomes, in total 44 of those chromosomes. Now, the 22 pairs of autosomes are identical in size and their homologous partner. So remember, we called them homologous pair, and we said they are having the same size, height, gene in the same locus, centromere in the same position. All 44 of them, the autosomes, or all 22 pairs, will have an identical size and everything that was mentioned about the homologous ones. The only difference will be the gonosomes. Now, the gonosomes in a female will have the same pair or the same size and everything else. Hence, we refer to them as XX chromosomes. However, in males, the gonosomes do not have the same size, do not have the centromere in the same position, and all of that. Let's look at what is happening here. With the female chromosomes, we can see that both these chromosomes are long. But in the male chromosomes, or gonosomes to be specific, we refer to them as X and Y. The X chromosome is the longer one, and the Y chromosome is normally short. So what you then end up having here is this chromosome will have other genes that will not be present in this particular chromosome because it is shorter. Hence, we refer to them as X and Y because they are different in size and the genes that they may carry. So XX for females and XY for males. This is a karyotype. The most common question that they will ask you when they give you a karyotype is for you to identify the gender of that particular karyotype. But before we rush, let's analyze this karyotype. So remember, we said we are able to see the number of chromosomes. So from both karyotypes, we should be able to get 23 pairs, meaning 46 chromosomes. Now, from pair number 1 up until pair number 23, let's focus on this one, the first one. So from this pair, we are getting 2 chromosomes. So that is chromosome 1 and chromosome 2 in pair number 1. And as you can see, these chromosomes are unreplicated. So you are able to see the chromosomes from each and every pair. Now let's see what is happening here. Last pair. The size of the chromosomes is different. One is longer and the other is shorter. So when they give you the diagram and they ask you to specify the gender of the human karyotype that you are given, you go to the last pair. So if the 2 chromosomes are different in size, then you say it's a male. But if the last 2 chromosomes, the gonosomes there, are the same in size and shape, that will be a female. That's all that you look at. Now, it is possible for you to be given a karyotype with an abnormality, meaning something that was not supposed to happen, but we'll speak about it when we do Down syndrome. So you will get to see an extra chromosome, maybe on chromosome number 21. So what you're able to see here is in each pair, there are 2 chromosomes. In our case, they are unreplicated. But if we were to have an extra chromosome here, that is an abnormality because each pair should have just 2 chromosomes. And here we end up having 3, but we speak about it at the end. So we are able to see that this human karyotype is of a male and this human karyotype is of a female. We focus on the 23rd pair. Then we looked at this, mitosis and meiosis. And I said, these are sibling topics. And you need to know the difference between the two. In mitosis, it occurs in your body cells, somatic cells, right? You damage your skin cells are going to divide to close off that gap, whether your skin is burnt or whatever. So whatever that is happening in your normal body cells, the cells are able to divide to repair that particular cell or a tissue. If we remind ourselves, we would also note that the cells that are produced in mitosis are identical, and the cells that are produced in meiosis are unidentical or non-identical. Now, when we look at the chromosome number remains the same in mitosis, and in meiosis, it doesn't remain the same, it actually gets halved. So in a cell that undergoes mitosis that has 8 chromosomes, it will divide only once to form 2 cells that will be genetically identical, which will also have 8 chromosomes each. The chromosome number is maintained. We started with 8 and we ended up with 8 in each cell. And there's only one division. The cell divides once and that is done. Now in meiosis, we say that the cell is going to undergo 2 meiotic division. That's the first one. And then there is the second one. Now what happens with the chromosome number here? The chromosome number becomes halved in meiosis. So if in gonads of a particular organism, there are 8 chromosomes, and that cell undergoes cell division, the chromosome number is going to be halved, meaning in each cell, we will have 4 chromosomes. Okay. Let's look at both of them. Let me try and zoom in. Mitosis, this is a parent cell which will undergo DNA replication. Now, these diagrams are very important. You need to be able to see exactly what is happening by just looking at the diagrams. So in this parent cell that is undergoing mitosis, how many chromosomes can we see there? There are 4 chromosomes. How many chromosomes can we see here? There are 4 chromosomes. What is different between the chromosomes that we can see on this diagram compared to the chromosomes that we can see on this diagram? The only difference is that the chromosomes in the first diagram are unreplicated. Then DNA replication takes place, and then you are able to still see 4 chromosomes, but in this case, they are replicated. Now, at the end of mitosis, we'll have 2 daughter cells. But now, look at the chromosomes that we find in our daughter cells. These are still 4 chromosomes in each cell and are unreplicated. Now, because remember in anaphase, this chromosome is going to be separated. This replicated chromosome is going to be separated, and this chromosome made up of one chromatid and this one made up of another chromatid will move to opposite side of the cell or opposite side of the poles. The chromosome number throughout is maintained. Now, in a meiosis division, so this is the same parent cell. And how many chromosomes can we see here? 4 chromosomes. And then the cell undergoes DNA replication. This chromosome made up of one chromatid is going to make a copy of itself. Then we have a cell that has 4 chromosomes before it 4 replicated chromosomes before it undergoes meiosis. Then what do we have at the end of meiosis 1? I'm writing here meiosis 1. We will have 2 cells. Let's look at the number of chromosomes in both these cells. We are having 2 chromosomes in this cell and 2 chromosomes in the cell. Already the chromosome number has halved. We moved from 4 to 2 and 2. But let's look at the chromosomes here. They are still replicated. Then these 2 cells are going to undergo meiotic division 2. I'm writing it as M2. Now, these 2 cells over here are going to be a result of this particular cell. Same with this one. These 2 are actually a result of this cell undergoing meiotic division 2. Let's look at the chromosomes there. Each cell contains 2 chromosomes. But how do the chromosomes look like? They are single-stranded or they are unreplicated. How many chromosomes did we start with? We started with 4. This is the 4. How many chromosomes did we end up with at the end of each cell? 2 chromosomes. The chromosome number is halved. So now the reason DNA replication occurs is for the genetic material to be shared amongst the cells. Because when this cell is ready, okay, I was going to say something else, but it will complicate things. But yeah, just to share the genetic material so that each cell can have the genetic material. But we know that here, the genetic material for each cell will be non-identical. So this will be non-identical, and that is due to prophase 1 and also metaphase 1 and 2, which we are going to look at. So if you ask yourself these questions, how many divisions does each process have? Mitosis has only 1 division. 1 cell divides once, done. Meiosis has 2 divisions. 1 cell will divide to form 2 cells, and then those 2 cells will continue to divide, 2 divisions. So in mitosis, there's only 1 division. In meiosis, there's 2 divisions. How many cells are produced? In mitosis, we're producing 2 cells. In meiosis, we're producing 4 cells. These are the differences. Now, remember when you are asked to write down differences, in the same point, you need to compare the same characteristic. So what do I mean by this? As much as this is written in a table, which you can be asked to tabulate, you follow table rules, you write your heading, you draw all your table lines, and you compare a similar characteristic in the same row. So from left to right, that is known as a row. Then from top to bottom, that is known as a column. So if we speak about the rows here, let's talk about them. So the first point speaks about location, where each process takes place. That is comparing the same thing. So on both sides in the same row, we are speaking about location. Mitosis occurs in the body cells, and meiosis occurs in the sex organs, which is the testes for males and the ovaries for females. It also occurs in plants, but we don't focus on plants in grade 12. Then, in the second point, we're speaking about the division of the nucleus and the division of the cytoplasm. Both karyokinases and cytokinases, this is the division of the cytoplasm, and this is the division of the nucleus. It occurs at once. But in meiosis, they occur twice. Why is that? Because of the 2 divisions. How many cells are formed in mitosis? 2 cells are formed. In meiosis, we form 4 cells. Now, how are the cells like? In mitosis, they are genetically identical. And in meiosis, they are genetically different, or you can say genetically non-identical. The word genetically needs to be there. What about the chromosome number? The chromosome number is maintained. It remains constant. And in meiosis, the chromosome number is halved. Then crossing over, we spoke about crossing over. Crossing over will occur in meiosis, and we don't speak about crossing over in mitosis. Now, when you're asked to compare, make sure to compare the same point in the same row. Even if you're not asked to tabulate, you will say mitosis occurs in body cells, while meiosis occurs in sex organs. We are able to read that sentence into having those 2 points for comparison. This I'm going to skip, is not important. This I'm going to skip, is not important. Now, we also looked at the revision for mitosis. Wow, I've been talking for close to an hour and I'm not checking up on anyone. Guys, are you still fine? Thumbs up, or if you have a question, I can take your question. So I can pause a little bit and take your question. Okay, thumbs up. Okay. I've been talking. Okay. Phases of mitosis. Remember, I mentioned this last week and I said sometimes interphase is considered a phase and sometimes it's not considered a phase. But in order for you to remember, you need to obviously know that interphase does take place and that is where DNA replication will occur. So DNA replication will occur during interphase, whereby a chromosome with one chromatid is going to obviously end up having two chromatids due to DNA replication. Then I said PMET is what you need to remember. I pass MET. PMET, that's the order of the phases: prophase, metaphase, anaphase, telophase. And we said we don't indicate any numbers here because there's only one division. Unlike in meiosis, where you will have meiosis 1 and meiosis 2, you need to indicate the numbers. So what do we have here? Let me zoom in a little bit because this is nice information here. So in I for anaphase, I is for in between, the part of the cell cycle of the cell that is between the cell divisions. So interphase will need to occur before cell division takes place. Then prophase, P for preparation. The chromosomes prepare for meiosis by untangling and becoming clearly visible. So we have chromosomes that are visible, and in prophase, there is crossing over that takes place. But remember, there is no crossing over in mitosis. This crossing over will take place in meiosis. M for middle, the chromosomes move to the middle of the equator. A for anaphase, A apart. The chromosomes, chromatids move apart. It will depend which phase we are speaking about, and then choose the correct terminology, whether we're using the word chromosomes or chromatids. T for terminal, that is the last or final phase of meiosis 1 on meiosis 2, if we are speaking about meiosis. But in just mitosis, we'll speak about just telophase. There is no 1 or 2. Now I'm going to read through this because we covered it. In interphase, the cell is not dividing here. Please note it is just DNA replication. So what we are having here is unreplicated chromosomes undergoing DNA replication in order for them to be replicated, for them to look like that. Now each chromosome will obviously have two chromatids, a chromatid and a centromere joining them. Then in prophase, please make note, we are still covering mitosis. In prophase, this is where we are able to now see the chromosomes. Now, when we see the chromosomes, we are able to count them. And in mitosis, we are not speaking about chromosome pairs. We are just speaking about chromosomes. Now, remember what I said, for each phase, you need to know what happens in that phase. You need to be able to identify the diagram when you are given to say, this is the particular phase. You also need to know how to draw the diagram. So what we are able to see here is the nuclear membrane disappearing. We spoke about it, the nuclear membrane disappearing. And then we are able to see the centrioles. Remember, I spoke about this terminology, centrosome and centrioles. Now, the centrioles will move to opposite poles. As they move to the opposite poles, they produce or they form what we call spindle fibers or spindle threads. Now, another thing that does disappear, which we don't see it in the diagram, is the nucleolus. So, prophase, you're able to see the chromosomes, and there is a nuclear membrane disappearing, and then we are able to see the centrioles moving to opposite poles. They would not have reached both poles at this point. They are just still moving to opposite poles. Now, how many chromosomes can we see in our diagram? 4 chromosomes, and they are not in pairs, and they are replicated. Then we go to metaphase, M for middle. So in metaphase, I want you to see what is happening with the centrioles. They have reached the opposite poles. What is happening with the centriole in prophase? They have not reached the opposite poles. They were still moving. Then in metaphase, these chromosomes align themselves in the middle of the equator. Now, each chromosome is going to be attached to one spindle fiber. So they are held in the middle of the equator by the spindle fibers. So if you are given this diagram, the giveaway is the arrangement of these chromosomes in the middle of the equator. And they arrange themselves single. They're not in pairs. So this will be a pair. So if we say chromosomes are in the middle of the equator in pairs, this would be a pair. Just give me a second. This would be a pair. So they are not in pairs. This is chromosome 1 and chromosome 2. They are not in pairs. It is just one chromosome arranged next to another. That is very important. And again, here we're using the word chromosomes, chromosomes, chromosomes. Then in anaphase, A for away. Let us look at what is happening in the chromosomes here. So the spindle fibers, these fibers are going to contract. And when they contract, they are going to actually cause the centromere to split. So if I have my other pen here, so this is a replicated chromosome, remember? This is a replicated chromosome. So meaning these are my chromatids and they are attached together by a centromere. So remember the spindle fibers are attached to the centromere of each chromosome. So during anaphase, now the spindle fibers are going to contract. When they contract, they will cause a split in the centromere. And when the centromere splits, the chromatids are going to move to opposite poles because they are no longer going to be joined together. They will be separated. So this one will move up and the other one will move down. Check what we can refer to them as. We can refer to them as chromosomes or we can refer to them as chromatids because we are able to see that is a chromatid or a chromosome made up of only one chromatid. That is very important. Opposite poles. Don't write opposite cells or opposite sides of a cell. They're not going to give you a mark for that. Then last one, telophase. So what are we able to see here? We are able to see that chromosomes have reached the opposite poles. And then what is going to happen here? Look at the chromosomes. They are still 4, but they are unreplicated. Then this is at the end. Remember the nuclear membrane disappeared in prophase? It reappears here in order to surround or enclose these chromosomes. The spindle fibers will disappear. The nucleolus that disappeared in prophase will reappear. Now, the chromosomes will unwind back actually to form the chromatin network. But what is the key thing here? We're able to see 2 cells. This is cell number 1 and this is cell number 2. That is our telophase. What is happening over here? We are having the cell membrane constricting. So this is preparing to basically separate the 2 cells. So eventually this will become a cell on its own and this will become a cell on its own. So that is called cytokinesis or cytokinesis, division of the cytoplasm. Remember to stop me anytime. Guys, you're too quiet today. All right. Then this is the terminology difference between cytokinesis and karyokinesis. So karyokinesis is the division of the nucleus. So this is completing telophase. So remember, we once had one nucleus in one cell, and now we are having 2 cells, and each cell will need a nucleus, so that would have obviously divided. Same with cytokinesis. Cytokinesis is the division of the cytoplasm. The cytoplasm is that jelly-like substance that fills up the cell. So it needs to divide so that both cells may have the cytoplasm. And at the end of mitosis, the 2 cells are genetically identical. There it is. All right, that's it with mitosis. Meiosis now is known as a reduction division. Why do we call it that? Because the chromosome number is going to be reduced. It's actually going to be halved. So in mitosis, if you were to go back and look, we started with 4 chromosomes. So these are the 4 chromosomes that we started with. And they are replicated in the parent cell. And at the end of telophase, we have 4 chromosomes. But in this case, they are unreplicated. The chromosome number was maintained. But in meiosis, the chromosome number is going to be halved. Now, in your somatic cells, the diploid cells, a full cell, we are going to have two cell divisions. Now, these two cell divisions will allow us to form haploid cells, which we call gametes. So gametes are actually sex cells. So sperm cells for males and egg cells for females. Now, meiosis will occur in the sex organs. In males, it is going to occur in the testes. So in males, it is going to occur in the testes. And you're not allowed to use any other words or terminology here. So you can't call them balls or anything like that. We don't recognize that language, so you won't be given a mark. So meiosis will occur in testes in males. Then in females, it will occur in the ovaries. So we have one ovary on each side. So one on the left and one on the right. And we can't really feel or touch them from the outside. They are inside the body. Unlike with males, you are able to, well, touch and see or feel. That's it. But we'll speak about it in human reproduction. All of that is produced during a process called gametogenesis, which we will do. Now, in plants, I'm not going to emphasize this because it's not tested. In plants, meiosis will produce what we call spores, and that can still happen on the ovaries in order to produce an ovule and all of that. But don't stress about this. They're not going to ask you about it. So let's look at meiosis. Meiosis, remember what is the goal here? We are producing sex cells. We are producing the egg cells and we are producing the sperm cells. So what are we having here? We are having this parent cell. And how many chromosomes do we have here? Now let me get you guys. I need to hear your voices. I've been prating. Come. Even Mulalo is quiet. Mulalo, are you there? Hi, I'm worried.
Yes, I'm
Can
here.
you guys
I'm here.
even hear
I'm
me?
here. I'm
Mulalo,
here.
are you there?
We are hearing you. Yes.
Oh, because maybe, oh, wait, guys. Wait. My computer is muted. Maybe that's why I can't hear anything. Okay, guys, are you there?
Yes, I'm here.
Okay, thank you so much. Even you are quiet today. You are stressing me, guys. Huh?
I'm quite. I'm just listening. I just want to understand.
Okay. Remember to unmute any second, guys, and ask me questions. The whole time, my laptop was muted. So even if you're trying to speak to me, I was not going to be able to hear anything. I just checked it now, and I've just unmuted everything. Okay. So anyone can tell me, Tombi, I see your mic was unmuted. Tell me, how many chromosomes do we have here? Tombi Futi, how many chromosomes? Okay. Zanele, how many chromosomes do we have here? Or anyone, guys?
We have 4.
4 chromosomes. 4 chromosomes. Now, these chromosomes in meiosis are going to be in pairs. So we need to figure out this color coding system together. We could say that these red chromosomes are from, we can say, the mother, and these blue chromosomes are from the father. But this is not the normal pair. So let's look at what is happening in the next diagram. In the next diagram, we are still having 4 chromosomes. This one from the mother, this one from the mother, this blue one from the father, this blue one from the father. How do they pair? It will be one from the mother, one from the father. That's why I said this is not a pair. This is not a pair. Because we don't call them a pair if there are 2 chromosomes from the same parent. It will have to be 2 chromosomes, one from each parent. So exactly what we have here. So blue and red. Now what we are able to see here is, oh, okay, there's a little bit of color red color in the blue chromosome and blue color in the red chromosome. That will be because of prophase 1 crossing over. But let's leave that for now. So DNA replication will take place at the end of meiosis 1. We'll have 2 cells. Those 2 cells will undergo meiosis and then end up having 4 cells. This is cell number 1, 2, 3, and 4. Let's check the chromosome number. Each cell contains 2 chromosomes. The parent cell had how many chromosomes? 4. Chromosome number halved. These are the phases, right? Meiosis will occur in 2 divisions. Prophase 1 to telophase 1 will be meiotic division 1, meiosis 1. See how I did not include interphase there. I didn't make a mistake. Interphase is not part of meiosis 1. It occurs before meiosis takes place. Oops, sorry. And then in meiosis 2, we are having these phases: prophase 2, metaphase 2, anaphase 2, telophase 2. It's PMET, PMET. Now, what are we going to have here? In meiosis 1, we have homologous chromosome pairs that are going to be separated. So we will have the 23 pairs of chromosomes that we are going to separate. At the end of meiosis 1, we are not going to have pairs. Now, variation, which is genetic here, is going to be introduced, and it is introduced during 2 processes. One being crossing over, and number 2 is the law of independent arrangement. Now, the chromosomes that arrange themselves in the middle of the equator will not follow any order. It is random. And then the chromosome number is going to be reduced. We move from a diploid to a haploid. What do we mean by that? So if this cell has 46 chromosomes, at the end of meiosis 1, we should have half the number of chromosomes, 23, 23. Then in meiosis 2, chromatids of whole chromosomes are separated. So now we come in with a replicated chromosome, but at the end, they're actually going to be chromatids. Again, genetic variation is going to be introduced by metaphase 2, law of independent arrangement. Drawing of chromosomes, I've spoken about it. This is very important. Let's look at prophase 1. I want us to keep track. I think I did it nicely last week because we're able to compare what is happening, and you would be able to see more than one phase on the screen, but we can't do that this time around. I'm only able to show one phase at a time. Now, how many chromosomes do we have here? I need someone to tell me, and I don't want the same person answering all the time. I need us to be active, guys. Tando, how many chromosomes can we see here? I didn't hear.
6.
6 chromosomes. Thank you, guys. 6 chromosomes. Now, are these chromosomes in pairs or not?
In pairs.
Thank you so much. They are in pairs.
Yeah.
So if a question, now this is my question. How many homologous pairs of chromosomes can you see?
3.
2,
3.
It's 3.
3.
Okay, it's 3. I think if you said 2, you just made a mistake, right? How many homologous pairs of chromosomes? This is the first pair. And here, just to make things easy, we've just color coordinated them. The orange ones, we'll consider them one pair. The red ones, we'll consider them another pair. And the blue ones, we'll consider them another pair. So there's 3 chromosomes. How many chromatids can you count? How many chromatids?
12.
12.
12.
4, 6,
12.
8, 10, 12.
12.
Ah, you guys are on point.
2, 4, 6.
Okay. Prophase 1. You need to know how to draw this face. You need to know how to identify the face when you are given it in a test, when you're given a diagram. You also need to describe what is happening in that face. The most important thing that you can mention here is what you see. What can we see? We can see the spindle fibers starting to form as the centrioles move to opposite poles. We can see it. So that's a mark. You are able to also see that the nuclear membrane and the nucleolus are disappearing. That is a mark. And you are also able to see that these chromosomes are in pairs and they're lying next to each other to prepare for which process? Crossing over. That is a mark. So if they give you this diagram and they say, identify the phase, you're looking, okay, what is happening with the spindle fibers? Okay, what is happening with the centriole? They are moving to opposite poles. They have not reached the opposite poles. And then what is happening with the crossing over? They are in, sorry, with the chromosomes, they are in pairs, and we can see crossing over taking place. That is definitely prophase 1. Then they will ask you, give a reason why you are saying it's prophase 1. Nuclear membrane disappearing, centriole moving to opposite poles to form spindle fibers. Nuclear membrane, I think I've mentioned this, chromosomes are visible and they are in pairs, and crossing over is taking place. Okay, this is a normal diagram that you will get in a test. These dashed lines are representing the nuclear membrane disappearing. This is your centriole forming the spindle fibers. They are not in opposite poles. What do we mean by opposite pole? One should be maybe here and the other should be here, forming spindle fibers. That will be opposite poles. In this case, they are not in opposite poles yet. Then look at what they have done here in terms of the chromosomes. This is a homologous chromosome pair. In that pair, one chromosome is black and the other, or is shaded and the other is not shaded. What is this telling us? This is telling us that one chromosome is from the mother and the other chromosome is from the father. So that means in total here, we are having 4 chromosomes. In total, we are having 2 pairs of homologous chromosomes. Okay, then we need to remind ourselves of this important process that occurs during prophase 1, which we call crossing over. Please note, if you name the process crossover, wrong. So if you say cross over, wrong. Over, that is wrong. So you need to say crossing over to indicate that is a process taking place. Because crossover, they will ask, are you crossing over the road from one side to another? We don't know what that means. So that's what they're going to argue. Now we need to remind ourselves of how this process takes place. Remember, we are saying our chromosomes are in pairs and chromosomes are replicated at this particular place, at this particular phase rather. So now the first thing that is going to happen is the homologous chromosome pairs are going to lie next to each other. So it'll be one chromosome from the father, one chromosome from the mother, and they are a pair. Remember what makes them a pair? Same size, height, centromere, same position, gene in the same position. Then what happens here? At adjacent chromatids. Adjacent chromatids meaning the chromatids that are next to each other. These chromatids. You can't speak about these outer ones. The ones that are next to each other, these are the adjacent chromatids. Of the homologous chromosomes, they are going to cross over. At a point where they cross over, this point over here, we call it chiasma or chiasmata. So the first thing, they lie next to each other, and then adjacent chromatids cross over. When the adjacent chromatids cross over, they are exchanging the genetic material. So at that point, material that was in maybe the red one in this case is now found in the blue chromosome, meaning maybe we can say red from the mother, blue from the father. I'm not sure why I'm following that color, but yeah. Then this particular red chromosome from the mother now has genetic material from the chromosome from the father. So now these two chromosomes, or this pair of homologous chromosomes, will continue the rest of the phases with the exchanged genetic material. Crossing over, what does it result in? Let's understand the whole process. This is an exchange of genetic material between maternal and paternal chromosomes. Maternal, a chromosome from the mother, paternal, chromosomes from the father. How does the process take place? They normally ask you for 3 marks to describe the process. Describe how crossing over takes place. I need these words for you to get a mark. Homologous chromosome pairs lie next to each other. The tick is at the end. If you just say chromosome pairs, wrong. Which word did you forget? Homologous. If you say homologous chromosomes and you don't include the word pairs, wrong. These 3 words are married to each other. Married for life. Homologous chromosome pairs. Even if you want to start with the word pair, you can say pair of homologous chromosomes. That's a marriage. Then the adjacent chromatids. There's another terminology I can introduce here, but you can ignore it if you feel like it will, because now I'm going to say non-sister chromatids. But let's leave it. Let's use adjacent chromatids. Adjacent chromatids of homologous chromosomes. Now, in the second point, whether you mention pair or not, you're not going to be penalized because you would have mentioned it in the first point. They will cross over at a point called chiasma or chiasmata. That's the second tick. And then genetic material will be exchanged. They will ask you, what is the importance of crossing over? Is to exchange the genetic material. What does it lead to? It leads to genetic variation of the gametes. The gametes will be genetically different. That means the offsprings will be genetically different. That is why you can have 4 siblings which have the same mother and father and may look very different. Not necessarily behavior and all of those things, because that can be influenced by external factors, but we are looking in terms of body characteristics, appearance, blood group, and all of that. So this slide, super important. Then metaphase, we are maintaining the chromosome number that we started with. We started with 6 chromosomes. But what do we have here? Metaphase 1, M for middle. But it's important for us to note what exactly are we finding in the middle of the equator. What you see here is the important part that you need to mention when you are asked. Identify the phase, then you say metaphase 1. Then they ask you, give a reason why you are saying that is metaphase 1. This is because the homologous chromosome pairs line up at the equator. You are able to see it. Remember that marriage, homologous chromosome pairs line up at the equator, or the chromosomes are in pairs. This is a pair that is lining up. This is another pair, and this is another pair. So it's 2 chromosomes attached to the same spindle fibers. Where are these spindle fibers now? You are having these spindle fibers fully formed, and the centrioles have reached the opposite poles. That's what you have. So that is super, super, super important. Now, one thing that we need to note here, there is a specific law, and that is known as the law of independent arrangement. This law is explaining that since you are having the 23 pairs of chromosomes here, there is no particular order in which these chromosomes are going to arrange themselves. So the reason we are different is also because of this. The chromosomes arrange themselves in any order. It is independent. So you can have chromosome number 21 being the first one to arrange, as a pair, and chromosome number 7, chromosome number 8, the number is independent. The arrangement of the chromosomes. So if this is your chromosome from the father and this is chromosome from the mother, there is no particular order to say all the chromosomes from the father, please come on top, and then all the chromosomes from the mother, you are here at the bottom. There is no particular law or arrangement for that. It is independent. What is at the middle of the equator? The marriage, homologous chromosome pairs. That is metaphase. Then this is explaining the random arrangement of chromosomes. It can be in any order. That's what you need to know. Then we go to anaphase 1. Now remember, let me get more pens. Remember in metaphase 1, we had a pair of chromosomes. I'm going to use my pens and I hope you guys will be able to see all of this business here. Yay. So remember in metaphase, we had this. So we had our pair of chromosomes arranged in the middle of the equator. Now, what is then going to happen in anaphase 1 is that spindle fibers are going to contract. When they contract, they will pull the whole chromosome. Remember in meiosis, we said a chromatid may be pulled. Now, a whole chromosome, my red chromosome with 2 chromatids will be pulled out. My blue chromosome with 2 chromatids will be pulled down. So the whole chromosome is going to be pulled to opposite poles. Now we can even see the shape of the chromosome changes. They normally have a V shape to show that they are being pulled. So the ones that are being pulled up is the 3 from the pair are going to be pulled up, and the other 3 is going to be pulled down. Remember I said opposite poles, not opposite sides of the cell. That is all that happens in anaphase 1. Then in telophase 1, you will then have the whole chromosome reaching the pole. Remember it was pulled by the spindle fiber and then they will reach the pole. So these 3 chromosomes that were pulled up will reach the pole, and the other 3 that were pulled down will also reach the pole. That's what we are able to see. Here at D, we can see the cell membrane constricting. That is telling us that there is a division of the cytoplasm. Eventually, we are going to have 2 cells. Now, what we need to note here is this is the end of telophase, and then we have 2 cells. Now, these dashed lines reappear, and these are our nuclear membrane. It reappears. Remember, it disappeared in prophase 1, and you're having 2 cells here. Now, the chromosomes do not uncoil back to the chromatin network. These 2 cells are going to continue with meiosis and undergo meiotic division 2. So we covered prophase 1, metaphase 1, anaphase 1, and telophase 1. We started with 3 homologous pairs, right? And we had 6 chromosomes in total. Now, this is what we had. We had 6 chromosomes in total, and at the end, we are now having 2 cells, and each cell has 3 chromosomes, and these chromosomes are still replicated. So that means those pairs are now separated. They are no longer in pairs. Prophase 2. Those 2 cells that we formed in anaphase 1 are now continuing with meiotic division 2. I hope we can see the difference. How many chromosomes do we have here? 3 chromosomes. Are they in pairs? No, they are not in pairs. The same thing will happen in terms of the centrioli moving to opposite poles to form the spindle fibers. Now we can see the chromosomes. These are the same chromosomes that we saw in telophase 1. So what are we able to see between prophase 1 and prophase 2? In prophase 1, we had 3 We have another one and another. Should have used a different color, but anyways. This was pair 1, pair 2, pair 3. This was prophase 1, and we had 6 chromosomes. In prophase 2, we are having 3 chromosomes, and they are no longer in pairs. 1, 2, 3. Now, I like it when I draw it like this because they're the same phase in just 2 different meiotic divisions. Meiotic division 1, prophase 1. Meiotic division 2, prophase 2. Let's look at the chromosomes. 6 in prophase 1, 3 in prophase 2. In prophase 1, they're in pairs. In prophase 2, they are not in pairs. Please note that in prophase 2, there is no crossing over that takes place. No crossing over. Then metaphase 2. Ooh, I need us to look at this. Still, we are finding chromosomes in the middle of the equator, but are they in pairs? No, they are not in pairs. Still though, these single chromosomes will arrange themselves randomly by the law of independent arrangement. Single chromosomes arrange themselves randomly in the middle of the equator. If we're to remind ourselves, in metaphase 1, we had something like this. We had a pair of chromosomes in the middle of the equator. So this was one pair, and this was another pair, and this was another pair. That's it. So let's see the difference, guys. So in metaphase 1, our chromosomes were aligned in the middle of the equator in pairs. This is a pair, 1, 2 chromosome, 1, 2 chromosome, 1, 2 chromosome. But in metaphase 2, it's only 1 chromosome, 1 chromosome. They are singly arranged. Please take note of this. Then anaphase 2. Also, the terminology that we used. Remember, in metaphase 1, we said homologous chromosome pairs align in the middle of the equator. In metaphase 2, we are saying single chromosomes align randomly in the middle of the equator. Then in anaphase 2, the centromere is going to split, and that centromere splits when the spindle fibers contract. Now, remember now we are having our chromosomes like this. It's just a whole chromosome that was arranged singly in metaphase 2. When the centromere splits, this is the same as metaphase of mitosis. The centromere will split, and then the unreplicated chromosomes, or you can say just chromosomes or chromatids, move to opposite poles. The terminology is important. Let me remind you of how the diagram was in anaphase 1. So in anaphase 1, we had the whole chromosome being pulled to opposite pole. So this was the whole chromosome, and that's my spindle fiber. This is another whole chromosome. My chromosome looks like hearts, but I'm not sure why. That's another chromosome. Then this side, we had another chromosome. Another chromosome. I'm writing very fast. Maybe that's why there's a whole mess here. Please, I need you to look at the diagrams, guys. Look at the diagram for anaphase 1 and look at the diagram for anaphase 2. What is being pulled to opposite poles in anaphase 1? It's a whole chromosome. It's a replicated chromosome. It has 2 chromatids. Why? Because it was initially a homologous pair. We separated a pair. Then it was a whole chromosome up, whole chromosome down. Then in anaphase, since the chromosomes were arranged singly in the middle of the equator, it will be a chromatid moving up, another chromatid moving down. I hope you can see this. This is a whole chromosome with 2 chromatids. This one that is being pulled now in anaphase 2, it is a chromosome with 1 chromatid. The terminology becomes important. Then telophase, cytokinesis, karyokinesis is going to take place. These are our unreplicated chromosomes or chromatids reaching the opposite poles. There is the end. 3 chromosomes in a cell, 3 chromosomes in a cell. The total is 4. Whatever that I was showing you here was what is happening in one cell in meiotic division 2, which is the same thing that will happen in the second cell of meiotic division 2. So what did we have? We had our parent cell, which had 6 chromosomes. At the end of meiotic division 2, there were 2 cells that had 3 whole chromosomes each. They continued with meiotic division 2, which ended up having about 4 cells in total with 3 chromosomes in each cell. The difference between the chromosomes that we find here and the chromosomes that we find there is because actually this, I'm talking now. I'm messing up. The difference between the chromosomes at the end of meiosis 1, the chromosomes are still replicated. But at the end of meiosis 2, the chromosomes are unreplicated. So I don't want us to be confused by the numbers here. Why is meiosis important? It produces haploid gametes. It produces the sex cells and the sperm cells. So if we're actually looking at meiosis taking place in human beings, okay, I need you to unmute here so that you can help me out with my chromosome numbers. Let me have something here. So 46 chromosomes in a human cell. Then the first meiotic division will take place. I'm not sure why the slide is moving up and down. And then the second meiotic division. I'm about to ask you guys questions, so please be ready to unmute. Okay, we are focusing on this, and this is taking place in a human sex organ. Remember, an organ is made up of cells. So we have 46 cells as human beings. Now, how many chromosomes do we expect to have at the end of meiosis 1 in each cell? How many chromosomes do we expect to get at the end of each cell? At the end of meiosis 1 in each cell. Sinako, how many? Okay, Zanele?
It's not 4, man.
Eh?
It's not 4.
Is it not 4?
Mm.
Okay, let's look here. We are saying the cell that is going to undergo meiosis has 46 chromosomes. As human beings, our cells have 46 chromosomes. Meiosis 1 takes place. How many chromosomes do
23,
we
ma'am.
expect to have?
23.
It's
Oh,
23.
23.
Yes.
Yeah.
Okay.
Are you
That
guys,
means-
why are you waiting for someone to say it and say it?
Okay, maybe other people are thinking, maybe my answer is wrong. Don't be shy, guys. Please do say your answer. Hey, Mulalo, please do say your answer. Don't be scared. If it's wrong, we rectify it. We're here to learn. So at the end of meiosis 1, each cell is going to have 23 chromosomes. I'm going to ask again. At the end of meiosis 2, we are going to have 4 cells. Please tell me, how many chromosomes are we going to have in each cell at the end of meiosis 2? Anyone? Okay. Tando? How many chromosomes in each cell? Okay. Anyone, guys? Anyone can answer?
23.
23 chromosomes. 23, 23, 23, 23 chromosomes. Okay, I'm happy to take questions. People were quiet, so I'm not sure if they were quiet because they were not sure of the number of chromosomes or they were quiet for whatever reason. I'm happy to take questions. Let me actually pause. I've been speaking for 1 hour, 36 minutes. I'm pausing to take 2 questions. There's no way that you don't have a question after that long presentation. So I'm listening. Any 2 or 3 people?
Hi, ma'am. I have a question.
Yes, Sanele?
So
Oh,
basically, I
Lilo.
wanted to ask, like, what's the like whole understanding or meaning behind daughter cells? Like, why is it daughter cells?
Why is it not, what is it, sun cells?
Like, why is it daughter cells, Mama?
You know what? Personally, I don't know. I'm not sure why they're using female words because even with chromatids, we have sister chromatids and non-sister chromatid. I must actually look it up, but I actually don't know the scientific reason. I'm not even sure if there is a scientific reason why they're saying it's daughter cells, or maybe it's because the female gives birth to, but it can be a son or a daughter. I don't know. I don't want to give you the wrong answer. I don't know.
Thank you, ma'am.
Okay, that was Lilo, right? I just saw two names. Okay, Lilo. Okay, anyone else, guys? First question down. I said I'm taking 2 or 3 questions. Zanele, do you have a question?
No, ma'am.
Okay. Anyone else with a question, guys? You need to have a question. There's probably something that you need me to clarify or something that was a bit confusing. 1 hour, 37 minutes. Anything?
Yes, ma'am. Is there a possibility of being asked to differentiate between a single phase? Say, for instance, asked to differentiate between anaphase 1 and anaphase 2.
There is a 99.99% chance of that.
Oh, okay.
Yeah, they can specify any phase. I should have a table somewhere on my notes where they can ask you to differentiate between prophase 1 and prophase 2, or metaphase 1, metaphase 2, or anaphase 1, anaphase 2, telophase 1, telophase 2. Oh, they can say differentiate between anaphase 1 of meiosis and anaphase of mitosis. Anaphase 1 of meiosis and anaphase of metaphase.
Okay.
So there's a 99.99% chance. I should have a table somewhere. But yeah, it looks like it's not in the next few slides, but I will show you that table. All right, I'm happy to take one last one if there is. Okay, let me continue. I know this is a lot, but yeah, let's please hang in there, guys. The terminology is quite hectic here. So why is meiosis important? It reduces the haploid gametes, or produces the haploid gametes. That's what I wanted to clarify when I did this example of 46 chromosomes and then 23 chromosomes in each cell. It halves the chromosome number. It introduces genetic variation. That's the main reason that if we speak about human beings, we then have siblings of the same mother and father, and then they would look different. Sources of genetic variation. The reason there is variation, one, it's because of crossing over. Two, it's because of the independent arrangement of chromosomes. Sorry, in metaphase 1 and metaphase 2, random mating partner selection is random. There is no particular order. Random fertilization, any sperm can fuse with any egg, right? And they will be genetically unique. So we will speak when we do human reproduction, and I'm not sure, I think I'll probably cover it a little bit when we talk about gametogenesis, because now the understanding is there's one cell dividing to form 4 cells. And those 4 cells are the ones that are going to grow and become sperm cells. But during ejaculation, we speak about males ejaculating millions of sperm cells. So where is all of that coming from? We do explain it in human reproduction. Then in females, the misunderstanding is, okay, a female releases 1 egg per month. But when we speak about meiosis, we are saying there are 4 cells at the end of meiosis. So what happens to the other 3 cells since a female only releases 1 cell a month? And where does this whole meiosis take place? So we do explain it in human reproduction. Now, these are the diagrams I'm trying
Ma'am,
to, yes,
sorry,
Sego?
ma'am, sorry to disrupt you. I just wanted to ask, but then you will continue. I didn't want to disturb you. So, are we gonna do a correction about the question that you sent last week, or because I have a question on the question on that question that you sent us. I have a
Okay.
question. So
Okay.
I wanted to know because you were teaching us about uh tighties, not balls, right? If you still
Yes.
remember that, not
Testes,
so
not
long ago.
balls. Yes.
Yes. So in the 2.2.8, right? There was a question there. It was asking about. I think it wanted us to differentiate between woman and female and male. So where on my side on the A I wrote sperms, and then on the B I wrote ovum, which is I feel like because I was not yet teached about this thing. I think it was tighties, not sperms. Or is that correct? Or you can go there when you answer the question.
Okay, I will check it. So I realized that there is a lot of information that we could not cover last week. So that's why I then used the slides for this lesson to cover the important part, because meiosis is covered over 12 hours at school. So we only did 2 hours. So I wanted us to make sure that we cover all the different aspects of meiosis before we mark. So we are going to mark. I am going to look at the question. So I think you're saying 228. State one place where meiosis takes place in males. The answer there
Yeah,
is testes. That's where it takes place. Then in
yeah,
females, it will be ovaries.
that's where I wanted to understand, because now when you're teaching us, you told us about titties, not balls, and then I wrote sperms. So now I understand that sperms come from titties, right? Not
Yes.
Yeah.
Yes.
Okay.
Okay. So this
Thank
is
you.
the understanding. So that's a good question. So in males, meiosis will take place in the testes to produce sperm cells. So sperm cells are produced as a result of meiosis. Then in females, meiosis will take place in the ovaries to produce an ovum. So yeah, that is that. But when we do mark, I will clarify everything. Okay, so I'm going to skip these diagrams and these diagrams. There was a question, I think it was from Samuel. So this is your slide, Samuel. So we are able to see what is happening here between meiosis 1 and meiosis 2. There should be another table that shows, I'll look for it, that is specific in terms of the phase. But yeah, we can still use this one. So chromosomes arrange at the equator of the cell in homologous pairs. So what phase would be this? So which phase do you think they are comparing here? Tell me. Chromosomes arrange at the equator in homologous pairs, and chromosomes line up in the equator individually. Which phase are they comparing here? Anyone? Anyone? Maybe let me come back to you, Samuel.
Meiosis two, I think it's metaphase.
Chromosomes line up in the middle of the equator. So this is a point for metaphase. Do we just say metaphase?
Metaphase 2.
Thank you. That is correct. That would be a point for metaphase 2. And here, that will be metaphase 1.
1.
So this is the difference between what is happening, right? Then I
Yeah.
need someone else. Whole chromosomes move to opposite poles of the cell. On the other side, chromatids move to opposite poles of the cell.
It's
Which phase are we comparing here?
anaphase
Lilo?
1 and 2, ma'am.
Phase 1.
Thank you. That's what I like to hear, guys. So this side will be anaphase 1, and then this side will be anaphase 2. My pen is acting up now, but I hope you can see. Then 2 cells are formed at the end of this division, and then 4 cells are formed at the end of this. So which phase here? Telophase.
It's all of
Telophase 1.
Thank you. Telophase 1 and telophase 2. Chromosome number is halved during meiosis, and chromosome number remains the same. So here we are just comparing the 2 divisions and not necessarily a phase. But let's look at the last one. Crossing over takes place. Crossing over does not take place. Which phase are we looking at here?
prophase.
Prophase
1.
1 and
Prophase 2.
prophase 2. Yay. Okay. Now, okay, maybe I'm moving too fast, but that is the different phases for each division if you were to be asked to differentiate. What are the similarities between these 2 processes? Both of them include the nucleus dividing. Both of them begin with one parent cell that has a diploid number of chromosome, a full number of chromosome. Both of them result in new daughter cells. The only difference is the number of daughter cells that are produced. Meiosis, 4 daughter cells, and then mitosis, 2 daughter cells. Both of them include multi-stage processes: prophase, metaphase, anaphase, telophase. Okay, there's no way we can get into this one. Let me see if I can answer one of the questions from that topic test. So just give me a second. Let's see if we can answer just one. This we'll have to do next week. Like I said, we can't just do 1 lesson or 2 lessons for meiosis. We do 12 lessons at school in order to cover, because it's 4 hours per week and we do it over 3 weeks. And also to mark activity. So just give me a second. I'm going to just try and do one question from that topic test that I sent you, then we call it a night. Where is this topic test? Okay. Just give me a second. I'm going to project it now. Ooh, I like this one. Okay. Did you guys answer this topic test? Did we answer it or not really? Not really.
We did answer.
We did
We did
answer.
answer.
Okay.
Yeah.
Okay, I'm just waiting for the test. So let's try and answer this one. 2.2. The diagrams below represent 2 different. If there's anyone that didn't get the topic test, just after the lesson, send me a private message, or even in the group, I will resend the test. And if I'm delaying anyone that has the test, please do send it so that we obviously help each other out. So what are we having here? The diagrams below represent 2 different phases. 2 different phases of meiosis of the same cell. You have diagram 1 and diagram 2. Give the names of the parts. When I call out your name, please do give me the answer so that we can move forward. Even if you just look at the diagram now and then you give me the answer, please. We need the names of part A, B, and C. So let me get
Can I
a
answer,
few.
ma'am?
Yes, I can't see who's speaking. Is it Zanile?
Yes, I
Snako.
can answer.
That was Snako.
Okay, Snako. Okay. Yes, Snako.
All right. So in A, I wrote, ma'am, spindle fibers. Then B, I said it's a centromere. Then C, it's a chromosome.
So B, you said centromere.
Yes.
And then C,
I said it's a chromosome.
No.
C. Let's look at C again,
Centriole.
Sinako.
Can I answer, ma'am? It's not a
Yes.
centriole.
Let me see. I want to, let's see. Okay, yes, speak so that I can see whose mic is unmuted.
Is it not centriole? C
Centriole.
centriole.
Yes.
Centriole. C centriole.
Centriole. Not a chromosome.
B, right?
Where is Sinako?
Yes.
Sinako, I want you to look
Yeah.
at this again.
Oh, C. Okay, now I see it.
Okay.
Yes.
So
Sorry.
can I just mention something? For C, you will get a
Yes.
mark if you wrote a centriole. You will also get a mark if you wrote a centrosome. Remember, one
Alright.
produces the other. And from the diagram, we can't see which one it is. Is it the one that is being produced or is it the one that is an organelle? So we take both. Okay.
Mhm.
Thank you, guys. I love the energy. Identify the phase represented in diagram 1 and diagram 2. Who's ready to answer? Anyone?
I'm ready, ma'am.
Can I answer this one?
I'm ready.
Can I answer?
La,
I can,
number
I
18.
can,
Avukile bani.
I can answer that one.
Fairness
Okay, I think that was
can
Khutlelelo.
answer.
Khutlelelo asked first.
the first one.
Come, Khutlelelo, we're
You,
listening.
you're gonna get it
Ma'am,
wrong
let
if
them
you are just
answer. It was Valentine's
I can
Day.
answer,
Let
ma'am.
them answer.
answer. Give us answer.
Okay, I can answer. It's metaphase 1, and B is anaphase 1.
Okay,
Okay.
I want to listen
All right.
again.
So I disagree with
Wait.
you.
No. Let's wait. Let's give each other a chance. If there's anything I need to rectify, I will rectify, right? So let me just hear your answer again. So you're saying diagram 1, you're saying it's metaphase 1.
Yes,
Then diagram 2, what did you say?
it's
Anaphase.
anaphase 1.
Anaphase 1.
Mm.
Okay. Okay, can I get a different answer? Anyone else with a different answer?
Yes.
Yes, Mulala.
From my side, number A is metaphase 2 and anaphase 1, and anaphase 1 on number B.
Okay, why are you saying diagram 1 is metaphase 2 and why are you saying diagram 2 is anaphase 1?
Ish. Wasn't ready for this one, but okay.
Okay, your answers are correct. Diagram 1, that is metaphase 2, and then diagram 2, that is anaphase 1. I want us to analyze this. Let's look at the chromosomes. In diagram 1, the chromosomes are arranged singly in the middle of the equator. So the reason we are saying they're metaphase 2, it's one chromosome next to another, next to another, next to another. They are not arranged in pairs. Remember in metaphase 1, they will need to be in pairs. There will need to be one chromosome with another pair, and then another chromosome in another pair, and then another chromosome, but here we're just having 2. So if they looked like this would be metaphase 1. Maybe just imagine it like that. Just look at it like that. So if there are 2 attached to one spindle fiber, so they are in a pair, metaphase 1. Now they are no longer in a pair, so that is metaphase 2. Then with diagram 2, this is metaphase, this is anaphase 1. Why are we saying it's anaphase 1? Because the whole chromosome is now being pulled to opposite poles. Are we fine, my people?
Yes, we're fine.
We are fine. I am
Yes, ma'am.
happy for people to make mistakes in the lessons and also in the test. You can make as many mistakes as possible. The nice thing about making a mistake when you are doing an activity is when you mark, you tell yourself, oh, okay, this was my mistake, and then you recognize it. You then make sure that in the next activity or next test that you do, you will make sure that you avoid making that same mistake. So that's why I'm saying I'm happy with you guys making mistakes now so that we don't make mistakes when we answer that exam. Okay, I'm running out of time. Well, name the phase that follows. Now, I want us to not only focus on what we did last week, everything that we've covered here, even if you had an answer, just recheck if your answer is correct or not. Name the phase that follows the phase in diagram 2. Let's raise hands so that I can see the hands and then choose a person. So name the phase that follows the phase in diagram 2. Remember, we said diagram 2 is anaphase 1. So you then need to tell me which phase comes after anaphase 1. Lebohang?
Prophase 1.
I didn't hear?
Telophase 1. Telophase 1, ma'am.
Telophase 1. That is correct. Telophase 1. If you did not write the number 1, wrong. Telophase 1. That 1 is important. 224 is the cell, might be cut off in 2 minutes time, I don't know. Is the cell in diagram 1 haploid or diploid? Please raise hands, guys, so that I can select. I think there's order when we do it like that. So please raise a hand so that I can select. I'm on, okay. I don't know how to, aneho. Mangale.
The answer is haploid.
Definitely haploid. Definitely haploid. It's half the number of chromosomes. State the function of structures A and B. Please raise your hand. Structure A is the spindle fibers. Structure B is the centrioli. I need the main functions. Please do raise your hand. I think I'm messing up your name. Aniaho or Mangale. I'm seeing the surname Mangale.
Yeah, you are correct.
The way I'm saying it is correct.
Yes, ma'am.
Oh, okay. Thank you. Please do give me the answers.
For which question, sir?
For this one, 226, state the function of structures A and B.
Okay, the function of structure A is the spindle fiber pulls the chromosomes to the opposite poles.
1 mark. Then B.
B, the function of structure B is the centromere the centromere connects to
Even if you use the word joins or
chromatids.
connects, just use the word joins though. It joins or combines the chromatids.
Okay, ma'am.
Okay,
It
spindle
joins the
fibers.
chromatids.
Yes, I'm listening. So the spindle
The
fibers
function of
function is to pull the chromatids or chromosomes to opposite poles. That's a tick. Then the function for the centromere is to join or combine the chromatids. That's the answer. Explain 2 ways in which meiosis is important. You can speak about the production of sex cells. You can speak about the introduction of genetic variation. I think all of that will be correct. State 1 place where meiosis takes place in males, that will be testes, and then in females, that would be the ovaries. I don't have time to answer the other questions, guys. So can we reserve this for next week. Is that fine? Because we've
Yeah.
done 2 hours already.
Yes.
Okay. All right. I really want to thank you guys for joining the meeting and always